1 second. There we go. I think that's the recording started. So if you want to start, Irene, off you go.
Okay. All right. Thanks, Ari. So hey guys, I'm Irene, a fourth year medic at GTC at Oxford. And this lecture is going to be, as you can see by the very long title, basically a whistle-stop tour of cells. So the organelles inside them, how macromolecules, particularly proteins, are moved within the cell and into the cell, how cells divide, how tissues grow, so through cell proliferation and cell growth, how we move from undifferentiated stem cells to more specialized somatic cells, and then how we look at cells with microscopy. And the format of the talk is we're going to go through each of those topics one by one with a little starter slide, which essentially has the Oxford first year syllabus on that area that you'd be expected to know. And then at the end, we're going to have a few multiple choice questions just to check you've been listening. And there'll be a few more at the end of the feedback quiz, at the feedback form, which will also get you the slides. So this is the first section, which is on organelles, which are the substructure within a cell. And throughout the presentation, the slides are going to be pretty word heavy. So don't worry about writing down everything I'm saying, just kind of listen and you'll get the slides at the end. So in terms of what's inside a cell, if we start with protein production, the proteins are produced in ribosomes. And I think one of my colleagues is going to give you a talk about protein production in more detail. But essentially, ribosomes have the large part and the small part. And mRNAs, which are produced in the nucleus, will come and be attached to the small part of the ribosome, and then transfer RNAs will attach to each individual mRNA. And transfer RNAs have a amino acid on the end, and then the large part of the ribosome will catalyze a peptide link between each individual amino acid to produce a protein. So that is the structure and function of a ribosome. And ribosomes can be free-floating in the cytoplasm, or they can be attached to the rough endoplasmic reticulum, which is what makes it look rough. And they're also found in mitochondria. So those are ribosomes. And then moving on to endoplasmic reticulum, these can be both rough and smooth. And so the rough is the bit that's attached to the nucleus, to the outer membrane. And that obviously makes sense because the nucleus is where the messenger RNAs are produced. And then the smooth endoplasmic reticulum is the bit that's not attached to the nucleus. And this is where membrane lipids are produced, and is also where macromolecules will go to be packaged into vesicles before they move to the Golgi apparatus. And the Golgi apparatus is where these proteins will be modified. So one of the most important modifications that you need to know about is glycosylation, which is the attachment of carbohydrate groups to proteins. And the Golgi is also where proteins are sorted and put into transport vesicles. So that is protein production in a nutshell, although we'll go into a tiny bit more detail in the trafficking section of this. So moving on to mitochondria, the most important thing you need to know is that there's the double membrane, as you can see in the little cartoon. And again, I think someone else is going to tell you about ATP synthesis in more detail, but essentially the basics that you need to know are that inside the matrix, which is essentially the cytoplasm of a mitochondria, is where the Krebs cycle happens, and the Krebs cycle produces NADPH, and then NADPH has to be oxidized back into NADP, which moves hydrogen ions out from the matrix into the intermembrane space, and then the electrochemical gradient produced by that between the outer and the inner membrane then allows hydrogen ions to move down through ATP synthase, which produces ATP. So that's the structure and function of a mitochondria, essentially. And a classic, MCQ thing is that mitochondria also have their own genome and protein production system, which obviously produces, when it goes wrong, can produce mitochondrial diseases. So that is important. And then looking at lysosomes and peroxisomes, these are both quite similar in terms of being single membrane vesicles, which are important in the breakdown of macromolecules. But the differences between them are essentially what they break down and how they break it down. And so lysosomes are found in all cells, and they contain lytic enzymes and an acid pH. And acid pH is important because the enzymes are made such that they will only work at an acid pH, which is important for compartmentalization, because essentially the cytoplasm of the cell has a neutral pH of around 7.2, which means if lysosomes accidentally split open and the enzymes are released into the cell, they won't work because it's not at an acid pH. So that stops the cell from accidentally digesting itself. However, when they're supposed to be breaking stuff down, the way that happens is that the things to be broken down are put into endosomes, which are just vesicles. And then the endosome fuses with the lysosome to form an endolysosome. And then because it's still at the acid pH, that breaks down, the lytic enzymes are able to work and break down whatever was in the endosome. So that is lysosomes, whereas peroxisomes are slightly different in that they're more specifically for fatty acid and amino acid breakdown. So they're particularly found in liver, which is where essentially all metabolism happens specifically, and also adipocytes because there's a lot of lipid breakdown there. And here oxidative metabolism happens, which essentially means that the breakdown of those two things often produces H2O2. And peroxisomes are particularly able to deal with this because they have peroxidases, which are able to break down H2O2 or hydrogen peroxide into water and oxygen. So that was a whistle-stop tour of organelles. And now in terms of the cytoskeleton, you don't need to know huge amounts of detail for this, but essentially it's good to know the different sizes, at least in terms of the order. So microfilaments being the smallest, intermediate filaments surprisingly being intermediate between them, and microtubules being the largest. And you should also know what each of the three are made out of. So microfilaments are made of actin. Intermediate filaments are different depending on what cell they are and have specialized structures and functions. And microtubules are made of tubulin, and they're important for intracellular transport, so moving organelles and macromolecules across the cell, and are also important in mitosis, which we'll talk about a little bit later. And then in terms of the nucleus, again, it has a double membrane, and essentially you just need to know a little bit about DNA structure. So the important things to remember is that DNA is essentially stored in nucleosomes, in terms of a nucleosome being being 8 histones, which are proteins, and the DNA wraps around the histones to produce a nucleosome, and the nucleosomes then organize themselves into the double helix that we're all familiar with. And DNA obviously has multiple roles, so it's both important for the actual transcription and translation to generate proteins, but it also needs to just be there and be stored so that it can be passed on in replication. And this is why we have both euchromatin and heterochromatin. And so euchromatin is like true chromatin. So So it's the bit that's being transcribed and then translated into proteins. And this is the one that's less condensed, which makes sense because when it's condensed, it's more difficult for the transcription machinery to essentially get access to the DNA. So when it's euchromatin, it's more spread out, which allows easy transcription, whereas heterochromatin is the storage mode, and that's when it's very densely packed, which then means that it's darker staining because it's denser. And when it's like that, then it's not being transcribed, or it's being transcribed much less. And in terms of heterochromatin, you can have two types of heterochromatin. So constitutive means it's never been expressed. And the classic example of this is telomeres, which are the bit at the end of proteins, of the chromosomes. And they're the bit that essentially protects it from wear and tear. And they get shortened throughout the cell life. And when it reaches a particular shortness, the cell will then enter senescence and will stop replicating. And then in comparison, facultative heterochromatin means that in some of the cell lineages it is expressed. And the classic example of this is the X chromosome. So in X inactivation, which is essentially the bit early in embryogenesis, one X chromosome at random will be inactivated because you only want to express, otherwise you just have extra DNA. And this happens randomly, which means that within a single female, some of the cells will have the maternal X chromosome and some of the cells will have the paternal X chromosome being expressed. And this is why we have the tabby cat image on the slide, because the reason that all tabby cats are female is that what makes them a tabby cat is that the X chromosome being inactivated, some of them, the cells that have it orange, is that, so there's the color is on the X chromosome, which means that in the cells where it's orange, and in the cells where it's black. And so because they have two X chromosomes, they have different colors, whereas if it's a one color cat, then it's male. And in terms of the nucleolus, this is the bit within the nucleus where ribosomes are produced. And so because ribosomes are where proteins are produced, the size of the nucleolus reflects how synthetically active the cell is. That is a nucleus. So there we go in terms of organelles. And we're now going to move on to trafficking, which is the word for transport within cells, essentially. So as a general rule, or in general, in terms of protein synthesis, the ribosomes that are on the rough endoplasmic reticulum is where the proteins which are destined to be either inserted in the membrane, secreted out of the membrane, or moved into lysosomes. So those proteins are produced in ribosomes on the rough endoplasmic reticulum, whereas proteins that are destined for the mitochondria of the nucleus are produced in free ribosomes. And the movement or trafficking of proteins from the rough endoplasmic reticulum, so the movement is from the rough endoplasmic reticulum into the Golgi network, where it's modified and sorted, and then those vesicles will move and fuse with the cell membrane, where they'll be either added there, so for example, membrane proteins, or exocytosed into the extracellular matrix. And you don't need to know about this in particular depth. I think you mostly just need to know that it exists. But we have signal sequences on proteins which determine where they end up and where they're trafficked to. And obviously, if you think about an amino acid, there's the N terminus, which is where there's the amino group. and there's the C terminus, which is where there's the carboxyl group, and the signal sequence is always found on the N terminus. So that's important to remember. And then in terms of secretion, this can be constitutive or regulated. And constitutive just means that it happens immediately. So as soon as the vesicle fuses with the membrane, it just releases its contents. And these are classically the extracellular matrix proteins because there's no reason for them to be stored. Whereas hormones are classically secreted by regulated synthesis because it will happen in response to a particular trigger. And so in insulin, in the beta cells in the pancreas, this will be produced and stored. And then when there's high glucose, that will cause the release of insulin. So that is an introduction to trafficking. And then in terms of movement into and across cells, so large macromolecules aren't able to just diffuse through the plasma membrane, but the way they enter is by receptor-mediated endocytosis, which essentially means that when they bind to the receptors, it causes a change in the membrane such that a bunch of adapter proteins gather, which then causes the membrane to indent into a clathrin-coated pit, and clathrin is just a protein on the inside. And this then, the membrane will fuse to itself to produce a vesicle, as you can see in the diagram. And this vesicle can then go into any pathway. So that is endocytosis. And then in transcytosis, then after that happens, rather than being used within the cell, it'll just move through the cell on the microtubules and then be exocytosed on the other side. And so this is classically in the capillaries where, for example, in digestion, things will move from the stomach into the blood and then into tissue. That is trafficking. And now the cell cycle. So this is essentially cell proliferation. I'm sure you have done this at A level probably. So mitosis is how we do classical cell division to produce an identical cell. And the stages, as you can see, are G1, which is where we essentially gather all the stuff that we need to divide. And this is the restriction point, which means that this is where you commit to division. And if you don't have enough of any of the materials needed to divide, you'll enter senescence or G0. But if you do pass this checkpoint, then you are going to divide. And after this, we do the synthesis phase, which is where we duplicate all the chromosomes. And then after that is G2, where we make all the proteins we need for mitosis. And then we do mitosis. So as you can see by this lovely diagram, these are the steps of mitosis. So in prophase, we start by condensing all the chromosomes. So they start off in the very diffuse stage of chromatin, which won't be visible, but now they condense into the actual structure of chromosomes. And during this phase, the nuclear envelope around the nucleus breaks down, and the centrosomes move to opposite poles of the cell. And this is important, the breakdown of the nuclear envelope, because the centrosomes, as you can see, are outside of the nucleus. But in order for division to happen, the centrosomes need to attach to the chromosomes. And this obviously can't happen if there's a nuclear envelope in the way. So then in prometaphase, which is just the bit in between prophase and metaphase, essentially that continues to happen. And mitotic spindles, which are just microtubules, which remember we talked about earlier. So the mitotic spindles attach the centrosomes, which are the bits that are at either end of the pole, and they start to build up towards the chromosomes. And then in metaphase, the chromosomes, they each have a centromere, which is the bit right in the center where the two like identical bits of the chromosomes, or the sister chromatids, are attached to each other. So that's what the centromeres are. And in metaphase, centromeres get attached to the centrosomes. And so then in anaphase, we essentially pull them apart. And this happens by the microtubules getting shortened because they are made of lots of different bits of tubulin. And so first we put more and more tubulin to make them longer, and now we're taking away the bits of tubulin to make them shorter and pull them to the opposite sides of the cell. And in telophase, that is completed, and then these bits, so the mitotic spindle can break down because it's done its job, and the nuclear envelope, which we talked about earlier, which had disintegrated and was just like lying around in the cell, then reforms to produce two individual nuclei. And finally, in cytokinesis, the cell actually divides to produce two identical cells. So that is mitosis. And in comparison, meiosis is how we produce gametes, so sperm and eggs. And meiosis, unlike mitosis, is a two-step process. So you start off with the normal G1 and S phase, so we now have twice the normal amount of chromosomes. And so let's quickly talk about diploid and haploid. So diploid just means that for every chromosome, you have two different chromosomes which have different genes on them. And so the first division in meiosis is the reductive division, which means that you go from diploid to haploid. And we'll talk about how that happens in a second. And so that produces two different daughter cells. So the reductive division is the division in which the genetic diversity is introduced. And then meiosis 2 is essentially mitosis. And so you then produce 4 haploid cells, of which 2 are identical to each other because mitosis produces identical cells. So we'll just look at meiosis 1. And so as you can see, in the differences in meiosis is that in prophase, when the chromosomes condense, crossing over or chiasmata happens, which is essentially where, so imagine we're going to say this is chromosome 1, this is the paternal and this is the maternal. When they get close together, there's then crossing over where the same gene, so we'll say gene 5 on the paternal and the maternal, are so close together that then when they eventually get pulled apart, the paternal gene 4 is put onto the maternal gene 4, and the maternal gene 4 is put onto the paternal gene 4. And so it's like swapped over. So that's how some of the genetic diversity gets produced. And then if you look at metaphase, whereas in mitosis, we'd have each of them lining up like all of the chromosomes are in a line, and you then separate the chromatids. Here, all of the chromosomes are in 2 lines. So you have chromosome 1 and chromosome 1 pulled apart, as opposed to the 2 separate chromatids being pulled apart. And so then what's important, another way that genetic diversity is introduced is by independent assortment. And this is essentially, as you can see here, this cell is getting, we'll say this is 1, 2, and 3, is getting the maternal chromosome 1 and chromosome 2, but the paternal chromosome 3. And that just happens randomly, which means that each of the different gametocytes that's happening in, there's going to be a different random assortment of chromosomes in it. So that's how some more genetic diversity gets introduced. And telophase and cytokinesis are essentially the same as in mitosis. So just going back quickly to meiosis 2, obviously in mitosis 2, you then have the normal situation where the chromatids are being pulled apart, and that's why they're identical to each other. So now we're just going to talk about differentiation for a bit. And differentiation is essentially, if we're thinking about embryogenesis, then we start with a fertilized like oocyte, and that oocyte then generates an embryo, which generates an organism that has a bunch of different organs in it. And so clearly during that process, we've gone from one cell that then generates a lot of different types of cells. And differentiation is the process by which that happens. So this involves essentially a lot of movement from euchromatin to heterochromatin, which we talked about earlier. So that only the proteins that are necessary for the structure and function of the individual cells in that tissue are being expressed, and the others are not being expressed. And terminal differentiation, or G0, happens either because the cell has reached its final state and it's like final differentiation, or because there's some problem in the cell, which means it shouldn't or can't divide anymore. So that is differentiation. And stem cells obviously are relevant to the conversation about differentiation. So stem cells are undifferentiated cells that are able to self-renew and generate more specialized cells. And you can have different levels of stem cells. So totipotent is the very first few cells after fertilization. And these are the ones that are able to produce not only all the cells of the body, but also the placenta. Whereas pluripotent is only able to produce all of the cells of the body. And multipotent are generally able to produce all of the cells within one tissue type. So hematopoietic stem cells are able to produce all the blood cells. Whereas unipotent means that they can only generate one particular cell type. But this is still different from a lot of somatic cells, which aren't able to replicate at all. And induced pluripotent stem cells are a recent-ish discovery, which is essentially that normal somatic cells which aren't capable of replication, for example, fibroblasts, so skin cells, can be de-differentiated back into cells capable of differentiation into many different lineages through the introduction of these specific transcription factors. And this is very useful, particularly for research, but in the future, potentially for therapies, because you can then push these pluripotent stem cells into a range of different lineage pathways. So, for example, producing neurons or producing other cells that aren't normally able to replicate themselves. So that is why that's useful. So now we're just going to go through tissue growth. And in terms of that, when we talk about cell growth, we're specifically usually talking about an increase in cell size. Whereas when we're talking about proliferation, we're talking about the cell cycle. And these are normally coupled in tissue growth, although there are examples where this isn't the case. As I've shown, that is the case in early development, where you're proliferating but not growing. And in terms of morphogenesis, as I've shown, and I think someone will be talking to you about embryology in more detail, but essentially that is proliferation and apoptosis, which is organized cell death, as well as differentiation, which allows you to go from a fertilized ovum to an embryo. And after birth, so during embryogenesis, most of the tissues will obviously be proliferating because they're growing. But after birth, most tissues will not be able to divide, which means that when there's cell death, they're not able to compensate for that. However, some tissues, particularly the skin and the gut, where there's constant loss, because obviously the skin we're always damaging, sloughing it off and burning, etc., then they have continuous division to compensate for that. And the liver is a very good example of a resting tissue, which is where there isn't continuous division, but when there is damage, it's able to compensate for that through division. And this is why we can have living donor liver transplants, where half of someone's liver will be taken and given to someone else, and then in both the donor and the recipient, the liver is able to replicate to generate a liver of the appropriate size for that person. And yes, so normal tissue will have balanced growth and death. And then in terms of increased growth in tissues, this can be hypertrophy, which means that there's growth without necessarily proliferation. And so obviously this will normally happen in non-dividing tissues which aren't able to proliferate, and can be physiological, for example, exercise increasing the size of muscles, or pathological, for example, hypertension causing cardiac hypertrophy and like left ventricular hypertrophy. And then hypoplasia is an increase in cell number through proliferation, and this tends to be physiological. So it's in response to there being, for some reason, insufficient cell numbers. So either a cut or something, meaning you need to generate more cells to compensate for that, or in altitude where there's not enough oxygen supply, and detecting that causes increased production of red blood cells to increase your oxygen carrying capacity. And so hypoplasia is physiological, but in neoplasia, it becomes uncontrolled and not in response to a specific stimulus. And that is going to produce a tumor, unless it's, I guess, in like leukemias and stuff like that. But tumors can be benign, which essentially tends to mean that they're well differentiated. So the cells are similar to in the original tissue type. And these benign tumors will grow by expansion, which means that they're not invading into the healthy tissue. It's like there'll often be a capsule and there'll be a clear distinction between them. But just because they're benign doesn't necessarily mean that they're not having like bad clinical effects. So because they're well differentiated, if it's something like a hormone producing tissue, like the pituitary gland or something, then well differentiated benign tumors will cause an overexpression of the of that hormone. For example, in the pituitary, it can cause acromegaly through increased growth hormone production. And then in terms of expansion in the brain in particular, because that's a set, like the skull means that that's like a set size, then a large expanding tumor, even if it's not like malignant, will cause an increase in intracranial pressure and can cause death, as well as like a lot of symptoms. But in terms of malignant tumors, these become increasingly abnormal because when you're growing without regulation or proliferating without regulation, then all the signals that would normally cause you to enter senescence, for example, a breakdown in telomeres or other abnormalities within the gene, no longer are acting as a stop to proliferation. And so you tend to accumulate more and more abnormalities, which then also means that your differentiation or protein expression is also abnormal. And so they get progressively abnormal. And malignant tumors tend to invade the healthy tissue, so they don't have a clean border, and that also makes them more difficult to excise surgically. And if they spread into the tissue, then they can also reach the blood vessels and the lymphatics, allowing them to spread to distant sites. So that is neoplasia. And then in terms of decreased growth, this can be developmental, which means that it's never hit the right size, or progressive, which means it was once the right size and then got smaller. And so in terms of developmental, it can either be agenesis, which means that it didn't develop at all. And an example of this is if your kidneys don't develop, then that causes Potter syndrome, which is where you don't have enough amniotic fluid, because particularly in the second trimester onwards, amniotic fluid is majoritively produced through fetal urine. And so if you don't have kidneys, then you're not producing urine. And this can cause a lot of problems and often like premature labor. And then hypoplasia means that it does develop, but it doesn't reach the right size. And a lot of the sex chromosome abnormalities will cause hypoplasia of the sex organs. So in XXY, you have small testes, and in single X, you have small ovaries. And then in terms of progressive decreased growth, this can be physiological. So the thymus is where T cells are produced, and at puberty, this involutes, so gets smaller and potentially disappears, and that is supposed to happen. Whereas it can also be pathological. And so general, in terms of it's happening throughout the different tissues of the body, can happen if the nutrients you're taking in aren't balanced to the nutrients you need. So that can either be through reduced intake in starvation, or through increased requirements in malignancy, because since malignancy is characterized by excessive proliferation, then it understandably requires a lot of resources. Or it can be tissue specific, which means in many places in the body, but in a single tissue type, there's atrophy. So this can happen in osteoporosis for a variety of reasons, but like poor calcium intake, or like reduced vitamin D, or like genetic diseases. And then local means it's happening in a specific area in the body. And so, for example, after breaking your leg and it being in a cast for a long time, your bone and muscle will atrophy due to disuse. Ischemia means that the tissue isn't getting enough oxygen for it to do the metabolic things it needs to stay alive. So this happens in the brain post-stroke. And obviously, because the brain is one of those areas we're talking about that are non-proliferative, it's not able to compensate after this. Neuropathic is essentially similar to disuse, because if a nerve is injured, so it's not able to so the muscle isn't like being used because the nerve isn't sending the signal saying like, oh, your brain wants you to move this muscle, then that muscle will waste away. And idiopathic essentially is all those neurodegenerative diseases that we don't really understand. So like with Parkinson and Alzheimer's, and generally we think is something to do with the accumulation of different proteins like tau and like beta plaques. So that is atrophy. And we have reached the final bit, guys, so good going. So microscopy, in terms of light microscopy, this is obviously standard. And the important things for you to know is the definition of resolution. And so resolution is how close together two different points on a specimen can be while we can still see them as separate. And it's important to remember that this inversely relates to wavelength, because if you imagine a wave like that, then if 2 points are like within a single wavelength, then they won't be able to be differentiated. And so the shorter wavelength is, the better the resolution is. And at the microscopy, the light microscopy resolution, we can see like the large organelles, but not the structure within them, or small organelles like ribosomes. And light microscopy, obviously, theoretically, you can have live samples, but generally, the way we go about producing the specimen samples generally means that they have to be killed. So we will fix it with formalin, which cross-links the proteins, which stops them being broken down, and so like preserves their structure. Then we dehydrate it, embed in paraffin, which allows us to section it very thinly, which is important so that the light isn't reflected weirdly. And then we stain, which the classic one that you should know for exams is hematoxylin and eosin. So hematoxylin will stain your nuclei and your acidic structures purple because it's a basic dye, whereas eosin is an acidic dye and so will be attracted to and stain your basophilic structures, for example, proteins, which is why the cytoplasm stains pink. And then we're just going to quickly go through what fluorescence is. So, you know, go back to A-level chemistry and physics. So fluorescent molecules are ones that have an electron at a given energy level that is able to absorb a light at a specific wavelength that has the perfect amount of energy to move that electron up to a higher energy like electron level. And then when the electron moves up, it then doesn't have the energy to maintain that level, and it falls back down again, causing it to release a photon of a specific wavelength, which will be of a longer wavelength, so a lower energy than the one it absorbed. And the reason that we care about this is that it allows us to give only a specific wavelength of light, and then essentially use filters on the microscope so that we're only looking or like absorbing the particular emitted wavelength, which then means if we have stained our samples such that only the protein of interest is fluorescing, we can look at only that. So it just makes things easier to see. And we can use fluorescence in a bunch of different ways. So in immunocytochemistry, we are using antibodies to look at specific proteins. And as you can see, here's the structure of an antibody. And the way we do immunocytochemistry is with a primary and a secondary antibody. And so the primary one is the one that's specific. So here, the antigen binding site on the antibody is what gives specificity. And so the primary antibody will be specific to our protein of interest. Whereas the secondary antibody, what it detects on its antigen binding site is this heavy chain. And so then it's not specific, but it's attached to a fluorophore, so the bit that's going to fluoresce. And so if we look at this is an example of it. And here we have this disease where there's IgA deposits, so that kind of immunoglobulin, and that's what we're looking for. And then we'll have a bunch of primary antibodies which recognize the IgA, and so we put them on the sample. And then we put the secondary antibodies. And the reason we bother doing that rather than just having primary antibodies with a fluorophore attached is that it allows signal amplification, because many secondary antibodies will bind to a single primary antibody, which will bind to a single bit of IgA. And so it just makes it brighter. And so that's immunocytochemistry. Whereas fluorescence in situ hybridization is how we detect nucleic acids. And this can be used for karyotyping, which is when you look at the chromosomes. And so that's an example of this here. So here, each of the red dots represents a chromosome 21. And here you can see this is a cell from someone with Down syndrome, because there's 3 rather than 2. It can also be used to look at messenger RNA, which allows us to look at what genes are being expressed and to what degree. And another useful thing is that GFP, or green fluorescent protein, is a classic fluorescent molecule. And because we know the genetic sequence that GFP has, we can use genetic engineering to tag that on to a protein of interest. So for example, if we wanted to look at actin, like micro filaments in action, then we could tag it on to actin, and then you are able to live image a cell, and you can see the actin as it moves because you tagged it green. So that's a cool thing. And finally, electron microscopy is how we can look in more detail at cells. So rather than using light as like the thing that we're using, we use a beam of electrons, which obviously have a much smaller wavelength, which means they have a much better resolution. And so in transmission electron microscopy, we essentially do the exact same thing as light microscopy in terms of passing it through a specimen and then seeing what light is being absorbed. And sorry, so it's not exactly the same. And that has the highest resolution. And as you can see ultrastructure, which is the structure of organelles. So you can see all the cristae, and that's good. And then scanning electron microscopy, you throw the beam of electrons at the surface and then see what reflects back. And so it has slightly worse resolution, but allows you to see the surface. So here you can see a red blood cell surface, and you can see the biconcave disc. But the disadvantages are obviously it's much more expensive, it needs more expertise, and you have to stain things with heavy metal. And the electron microscopy is happening in a vacuum, so you can throw the electrons, and that means you definitely cannot do it with live specimens. And it also doesn't give you the opportunity to do different colored stains, because electrons don't have color, so it's just black and white. So there we go. If people wish, we can now do a Socrative quiz. So if people want to log on to the room, it's Matthias 7563.